So typically, if we're analyzing genetic variation of the human population, of human species, or let's say we're interested in, we'll see in a second, in analyzing a frog population, we typically don't want to analyze every single individual of a species. And so one reason for that is that, that's kind of impossible. The size of a population becomes so large that you can't possibly analyze all those individuals. But kind of more importantly is this idea that the set of populations of a species are not freely mating with each other. And so typically that's because there's some sort of geographical barriers that prevent mating. And this could be something like distance, where like an individual in California is unlikely to end up, especially if you're a frog, an individual in California is very unlikely to find themselves able to breed with an individual from Georgia. And so this could either be like distance, this could be things like mountain ranges, this can be things like oceans. And so there's some sort of separation that prevents this free mating from occurring. And that doesn't mean that that the mating is completely closed down. So, you can still have migration between different populations, but at some level you want to kind of restrict your analysis to individuals within a short geographical range, a small geographic area. And so at this point we want to measure kind of what genetic variation is present within that population, and then also we want to get a sense of the frequency, how commonly occurring a given allele is. And so we do this by having three really important measures. And so those are allele frequency and genotype frequency. And they can be related to each other, but typically you need to think about them as being able to be independent from each other. And so, for a given locus, we're looking at 1 out of the 3 million options right now. So there's 3 million genetic variations. We're going to restrict ourselves to looking at a single one. And so for this particular change, this could be an A or a C, for example. We actually analyze what alleles are carried by each individual within a population. So our population of frogs, and it's pond 1, so somewhere there's a pond that has 16 frogs. So if we counted how many frogs within there were 16. And so for all 16, we actually determine the genotype at a given locus. And so we could do this by DNA sequencing. If these alleles have an effect on some sort of phenotype, we can do that through the genetic analysis that we did in previous chapters. And so from this information, we want to summarize how many individuals have each of the 3 possible genotypes. And so that's what this is right here. So at a first level, we simply just count how many individuals are big A, big A. And so 1, 2, 3, 4, 5. And so we calculate 5 right here. And then how many are heterozygote? There's 8. And then how many are homozygous recessive? And so to calculate this as a frequency, we simply just need to divide each of these numbers by the population size. And so from here, we end up having an allele frequency of 5/16, 8/16, and 3/16. The allele frequency is similar, where you're just calculating the total number of big A's and little a's in this pond. And so we could also go in and for each frog calculate how many big A's are there. And so here, we've added up all of those together, and it's 18 big A's and 14 little a's. And so if you add these two numbers together, that would equal 32, and that is just equal to 2 times the population size. And so that's just because a diploid animal carries 2 alleles. So the diploid, you carry 1 allele from your mother and 1 allele from your father. And so if we wanted to get to allele frequency, we would simply divide by 32. So again, if you add up these frequencies, the sum has to equal to 1. So 30 seconds plus 30 seconds is equal to 1. And that's because, every allele has to either be a big A or a little a. And then for the genotype frequencies, your genotype has to be one of those three options. And we refer to the allele frequency. So this is an important definition. So little p is the allele frequency of the first allele, and we refer to Q as the allele frequency of the second. And so, one of the things that can be confusing at first is that we say there's a relationship between genotype and allele frequency, but they can be independent from each other. And I think that's easiest just to see if, let me get some space really quick. By comparing something like this. So you could have a population like this, or you could have a population like this. And so in this case, you have 8 homozygous individuals, 8 homozygous recessive individuals. So the total number is still equal to 16. And so, the total number of alleles of each that are carried would be 16. Or you could have a population where every single individual is heterozygote. So all 16 individuals are heterozygote and none of them are homozygous, either recessive or dominant. And so again, if you calculated the number of big A alleles, it would be 16, and the number of little a alleles, it would also be 16. And so these are two situations where the allele frequency is the same, but the genotype frequency is completely different from each other. So you would still calculate the allele frequency from the genotype frequency, but they aren't fixed together, if that makes sense. All right, so this hopefully introduces 4 important terms. Population, so this is a group of individuals that are analyzed together. They are all members of the same species, but they're not all of the members of a species, and they're geographically or some way restricted in area so that they're much more likely to mate with each other. The population size is just the number of individuals in that population. The allele frequency is a measure of how common a particular allele is. And then the genotype frequency gives you the distribution at that genotype levels. So, kind of to take what we've just done, we've calculated the allele frequency and the genotype frequency for a single locus. But it's important to remember, like humans, there's millions and millions of differences that exist. And so, here we have just looked at the allele frequency for a single one, and, it could be on chromosome 1, and it could be at position 364, and this might be an A to a C. And so we can calculate the allele frequency, and so 80% of the alleles are big A, 20% of the alleles are big C. But there could also be another locus that we measure. This could be on chromosome 2. This can be in position 6,342, and this might be a G to a T. And so again, we can calculate these numbers within that population of how frequent the G is and how frequent the T is. And so, we can keep doing this until we're looking at millions of different positions, and we calculate the allele frequencies for every single one. And we can do this for different populations. So this could be the set of frogs in pond 1, and then if we go to a second pond that's nearby, we can calculate these numbers in all of the frogs that are living in this pond. And, typically these numbers will be close together, but they won't be exactly the same. And so the reasons for that we'll get into in the part 4 of the lecture, but there's evolutionary forces that change the frequency of alleles. And so we might measure this as 80% in one population and 75% in another population. But there can be very profound differences. So if we look at this last one, this allele is 20 times more common in pond 2 than pond 1. And so, kind of this, just to belabor the point, you're calculating these numbers for specific populations, and these numbers do not necessarily need to be the same. Practically, this can be very useful for us because we can actually use this information to do things like assign ancestry. And so what we're doing is we're taking advantage of the fact that the allele frequencies from individuals from the British Isles are different from the allele frequencies of individuals from Poland. And so again, there's this geographical separation. Individuals that live in Poland are more likely to mate with individuals that live in Poland, especially historically. And so this causes, over time, to be a change in the allele frequency between these different populations. And so you can use this information along with various statistical algorithms to actually analyze your genome and determine your frequency of your distribution of SNPs is most consistent with individuals of kind of like which population. And so that's kind of how, for me, the ancestry basically determines the majority of my ancestry comes from the British and the British Isles. Most of my ancestors are from Ireland. 2/3 of my ancestors are from Ireland. And so that's kind of how my distribution of alleles is most consistent with.